Nikita Alexeev will present the talk about PAREBRIC, which is an acronym for Method for Detection of Parallel Adaptation with Genome Rearrangements in Bacterial Populations. And welcome.
Thanks so much. My name is Nikita Alexeev. I'm from Utmo University, and I'm presenting a tool, PAREBRIC, for identification. Actually, the acronym is for parallel rearrangement and breakage in bacteria. So, the motivation for our tool was a case from a quite recent paper where the authors studied Streptococcus genomes, and they found out that a particular genome rearrangement, which is shown here on the left, which inverted a part of a segment of a genome between PHTB and PHTD genes. This inversion happened simultaneously, independently in several strains. The strains where it happened marked with green circles on a tree. So, as you may see, they indeed happen independently. There is no explanation with just one common ancestor where it happened. So they happened just randomly in several places. So it can be the reason for that can be that it's this inversion gives some benefits, some biological benefits to these particular strains. And indeed, it was shown by another group of authors that this inversion actually helps the strains to avoid immune response. So our idea was to make this process of finding parallel rearrangements automatical. What exactly does it mean? It means that we color all the strains with a particular rearrangement. We color them on a tree, and we check if this coloring is consistent with a tree, which means that the color, the appearance of green color can be explained by just one evolutionary event, not several. So on the right, you see that there are 4 strains, but this coloring is consistent because the coloring is consistent because here, in common ancestor, it can appear actually, and all the strains from 4 to 7 just inherited it. While on the left, it's parallel events. This green color appeared independently at least twice, and we developed a method to do it automatically. So what does exactly I mean? I mean that as an input, the tool takes a collection of strains represented as a sequence of synteny blocks and their phylogenetic tree. And well, there is our method, which is black box here, and I will explain briefly what happened in this black box later in the talk. And then it outputs a list of rearrangements which happened parallel, arranged by their importance, and I will explain what does it mean later as well. So you give it a sequence of strains, you give it a set of strains, you give it a tree, and you get what rearrangement can be important biologically in this genomes family. Let me explain how we find which rearrangement happen in how we actually map the rearrangement information on a tree. So we start with synteny blocks. I already said this word several times. What does it mean? It means that just conservative genomic intervals, which are shared between several strains, and we have a collection of strains, each represented as a sequence of blocks. So as shown here, we color all the block ends in such a way that the adjacent vertices are colored the same color in a particular strain. Then we inherit the same color to all the strains. And then we check what happened with each particular adjacency. Adjacency is a pair of adjacent vertices, adjacent in strain 1. on this picture. So we are looking here, for example, of this red adjacency, red pair of red vertices, and we see what happened with this pair in other strains. So, for example, here in strain 2, we see that there was an inversion of the second block. So both the red vertices are neighbored with yellow vertices. So this is one type of event. In strain 3, we see that red vertices are neighbors with purple vertices. So it's another type of an event. It's an inversion of this sequence 2, 3, 4. In strain 4, it's not so easy to say what's exactly happened, but sometimes it was also the same inversion in strain 3, because red vertices and purple vertices are neighbors again. And in strain 5, there are different colors of neighbors of red vertices, which means that there was some complicated event. There was some complicated event during the development of strain 5. So this gives us possibility to say what exactly happened with a particular adjacency in all the strains, and according to this information, we colored them on a tree. So we say that all the colors are different, except for strain 3 and strain 4. For them, the color is the same; it's green, and the color of the tree according to this information. And we get such a coloring for all the adjacencies. And for all the adjacencies, we check if they're parallel or not. Again, parallel means something look like on the left picture, and on the right picture there is no parallel event. So if it's parallel, it may be interesting because it happened independently, so it may have some biological meaning for the strains to get this rearrangement. Okay. So then they do the same for copy number variation. Some blocks can be presented in some strains in different number of copies, or can be not presented at all. So, for example, here, so for each block, we just count synteny block. We just count how many times it appears in our strains, and we color the strains according to the number of blocks it has. So here, for example, strains 1, 3, and 5 are colored white because they have just one copy of our block 1, and strains 2 and 4 have two copies, so they color different colored blue. And again, we check them for we check all these patterns for being parallel. After this procedure, we have a number of colorings on the same tree. Some of these colorings are parallel, some of them not, and we believe that parallel could be more interesting. But moreover, we do some analysis on these colorings because not all the parallel, not all the inconsistent cases are important equally. For example, here you may see that actually both colorings here are inconsistent, but the picture on the left, well, it's probably just some errors in our input data. Probably phylogenetic tree reconstruction wasn't accurate enough. Probably something was wrong with genome assembly of a strain, of this white strain here in between two green ones. So this, formally speaking, it's inconsistent, and there is a parallel rearrangement here, but it may be not interesting. And on the picture on the right, at the same time, we have that this particular rearrangement, well, the particular arrangement which is shown here, it's appeared several times in all the different places, and this seems to be more interesting. In order to take this into account, we provide parallelism score for all our parallel rearrangements, which computes, well, I wouldn't explain in details how it computes, but it computes taking into account how many times each color appeared on our picture and how distant they are from, how distant are they from each other. And that gives us a way to say that this particular arrangement is the most important, and actually arrange them by their importance. And also there are cases when some patterns of some colorings are very similar for different blocks. For example, here, the same block, the block 87, has very similar pattern phylogenetic pattern with block 91. You may see here they're more or less the same. There is just slight difference. So we don't want to ask the researchers who work with output of our tool go through all of it. So we do some clustering, which takes into account both the phyletic pattern and the distance between the blocks in order to clusterize them. So we, after both this procedure, we have a nice list of genomic rearrangements, inversions, insertions, deletions, arranged by their importance and also clustered like if they're similar, we just see only one representative of such a picture, and how it may be helpful. For example, here we indeed we confirmed the case I started with from Pavel Shelyakin paper. We work with the same Streptococcus family, Streptococcus genomes, and our parallelism score showed that the most important one is indeed the one which is mentioned in the original paper, and we see all the strains which has it. So if our tool was there well, a year ago, it would save the authors some time and manual work. We applied it to Streptococcus pyogenes genomes. We find out there is an inversion between RNA operons, and this is the most parallel rearrangement for these genomes. We do not know what biological benefits this inversion gives, but it definitely, well, it's work in progress, and it's definitely worth to check why it's happened so often and so independent in such an independent way. Another way how it may be, well, this is how this operon, how this inversion between operon looks like. Another way how we can apply our tool is we applied it to E. coli and Shigella genomes, and we find out an insertion which actually distinguish whose pattern actually more or less the same pattern as which shows who are invasive strains and who are not. So if we do know the phenotype, we may find some genomic architecture features which may explain this phenotypical difference. And well, yeah, let me skip this since I almost don't have time. We have some visualization as well. So our tool is able to investigate, can help you to investigate the bacterial genomes on the level of genomic architecture, not only on the level of SNPs, but on the level of genomic architecture. And it may help you with finding what's the connection between this genomic architecture and phenotype. It's available on GitHub, so you're welcome to use it and send us your feedback. And also I want to thank my collaborators, which is Alexey Zabelkin, PhD student who actually did the most of computational work and programmist work, Yulia Yakovlev from St. Petersburg State University, and Olga Bochkaryova, who is also the next speaker from IST Austria, and all our funding, and thank you for your attention.
Thank you. Any questions? Sinan, I'm not sure what you have shown on the Shigella slide, one of the last ones.
Oh, let me try to.
So what? Well,
okay,
no,
it was
what was the recurring rearrangement that you've observed?
Actually, what we saw is that there is an insertion which actually it's presented in all the Shigellas and all the only invasive E. coli strain, and do not present it in
it's the insertion of this lipase gene.
Of this IPH. Yeah.
I see.
Yeah. I believe it was also known, but it's like it's the mathematical way to find the same thing, and we yeah.
Actually, it wasn't.
Oh, okay.
We are writing a paper about it. Okay. Well, then that's it. Thank you. Oh, sorry.
Yeah.
Yep.
Thank you for the election. I'm Viktor Mamontov. How do you think such rearrangements
Could you open your mask because
could they impact gene expression in bacteria?
Well, it makes sense to analyze what we analyze together with gene expression pattern, but what we do is only genomic architecture. It's a good idea to combine different approaches, but here we do only.
People wouldn't have transcriptomes for all these strains, but a short answer is yes. For instance, you may have a rearrangement reordering promoters, and it's a well-known mechanism of phase variation. Sorry for intervening, but eventually there is stuff that I understand. Okay, well, thank you.
Thanks so much.